In this lecture we are going to study string search algorithms. So before studying these string search algorithms, let's first define the different elements here. What are strings? A string can be defined as a sequence of characters, and it can be a constant or a variable. If we make a link between this concept and programming in general, we can have constants and variables, and a sequence of characters can be a constant or a variable. And the other thing is that we can relate these strings to a data type or structure, a data structure. So it will be similar in this case. Now let's try to define with more details what is a sequence of characters. In order to define a sequence of characters, we need to define what is a character. A character will be a unit of information. And the mathematical way to define this will be to say that a character will be an object belonging to a given alphabet. And the alphabet in this case is simply the set of possible characters. So as you can see, the string will be defined as a sequence of characters. So the second element of importance is sequence and characters. And a character will be a unit of information and is mainly defined by the alphabet, which is the set of all possible characters that may exist. Now, what is the difference, for example, between a string of a sequence? So a sequence is a string is a particular kind of sequence that is made only of characters. So a sequence is more general. A sequence is just an ordered collection of objects. So in this case, an object is just a mathematical term to say anything. Okay. And there is another concept that is related to the concept of string and sequence, that is the list. And what is the difference between a sequence and a list? The difference is that a list will be a sequence in computer sciences. For example, we can say a list of integers. And then a string will be more precisely a list of characters. Most of the time we will be dealing with these sequences of characters in computer sciences. So we can use the term list of characters here if we are really using an implementation of a string. Now let's try to give some examples. Let A1, A2, and so on be an alphabet. So A1, A2, and so on are the possible letters, the possible characters that we can use. Let S with S1, S2, and so on, Sn be a string. So here the size of S is simply n because we have n characters. And another important thing is that for all I, Si will belong to the alphabet. And we can also define the character at position I is the character at position I. And we can also define chunks of strings. So for example, we can say S between I and J will be simply Si, Si plus 1, and so on until Sj. So we call this chunk of strings a substring. And similarly to this definition of a substring, we can define a prefix, and we can also define a suffix. So the prefix are the first elements of S until position I, for example. So S from 1 until I will be a prefix. And the suffix is the last part of the string. So, for example, S, I until N will be a suffix of S. Now let's give another example, but this time not in terms of general concepts, but for the DNA string example. Okay. So in this case, A will be simply the set of characters that we can have in DNA. In DNA, most of the time we will say that we will have 4 letters. So we have letter A, letter T, letter C, and letter G for each one of the elements that may constitute the DNA. Now, a string, you can define your own string. Let's just make a little example. So we have a repetition of 3 letters A, then T, then C, for example, then another C. That's it. We have our string. In this case, the size of S is simply 6. And we can also define the prefix and the suffix. For example, the prefix until position 2 will be simply this part of the string. And the suffix from position 4, for example, will be this part. Okay, so this would be the suffix from 1 to 2, and this would be the the prefix, and this is the suffix from 4 till 6. The concept of string is pretty much defined, and we are going to focus now on the term algorithm. So what is an algorithm? We can think of an algorithm in terms of input and output. So this is very general, but in fact the definition of an algorithm could be quite general. So the thing that is going to allow us to go from this input to this output will be the algorithm. That can be defined simply as a finite and unambiguous sequence of instructions. So The algorithm is the sequence of instructions that will allow us to go from the input to the given output that we want. And it's quite general because we can, for example, say that a recipe that will allow us to go from the raw material, the raw food, for example, just flour, water, milk, whatever, we apply this sequence of instructions that are in the recipe and we obtain our cake, for example. So this would be an algorithm. Most of the time, however, algorithms are implemented in computers. Okay, for example, we have a bunch of numbers, we want to sort them. We will apply an algorithm to sort the different numbers. This algorithm is implemented in a computer, and the algorithm is just a sequence of instructions that will allow us to sort the different numbers. There are different ways to characterize an algorithm. So, characteristics of an algorithm. First, we want to know how well is the algorithm solving the task. So this is the quality of the algorithm. Is it solving the task in a perfect way, or just in a probabilistic way, or it has a given quality? Okay, this is a first way to characterize the algorithm. The second way will be its time complexity. We can write it O of something. So this something will be the variables that will be important in order to define how much time the algorithm will take. So for example, if let's imagine that here we have n square, and let's n square be the size of the string. So if we have an algorithm that is running in O of n square, it means that when we will have 10 more strings, for example, or our string is 10 times larger, the time complexity will be to the power of 2. Okay, this gives us an idea of how much time will it take to run the algorithm when the main variables increase. The third element here is the space complexity. We also write it in the same way, so it will be O of something. For example, this something here could be n to the power of 3. So it's the amount of space, of memory that we will need to run the algorithm. So here it means that we need to consider the size of the string to the power of 3 in terms of space. And these two things will be very important, particularly in bioinformatics, because in bioinformatics, the n here, the size of the string will be very, very large. So if we have like huge powers here, not even huge, but even to the square, we may start having issues with the time complexity and the space complexity, as we will see later on. So in a nutshell, the algorithm is just this finite and unambiguous sequence of instructions that will allow us to go from an input to a given output. Most of the time, algorithms are implemented in computers, and in order to characterize an algorithm, we have 3 elements to consider. First one is how well the algorithm is solving the task. The second element is the time complexity, and the third element is the space complexity. So how much time is the algorithm going to take with respect to some important variables, and how much memory the algorithm will need to run with respect, again, with some important variables. If we want to understand in a deeper way what is time complexity and space complexity in computer sciences, check out the corresponding video. So in order to include the term search and have now a definition of string search algorithms, let's just take another piece of paper and let's work on this now. So I will rewrite string search algorithms and let's go to the definition. So let A be the alphabet. So we have seen through the definition of a string that a very important element is the alphabet here. So here we define the alphabet. Let S be a set of strings such that for all S that belongs to S, for all Si in S, Si will belong to A. Okay, so S here, this special S is the set of all possible strings considering the alphabet. And now we are going to define a function f that is going from this S square, so it's taking two possible strings, and it's going to 0 or 1. So this function is taking two strings and it outputs 0 or 1. Such that for two strings S and T, f of S and T will output 1 if S is a substring of T, and it will output 0 otherwise. So just a brief recap. We have defined the alphabet, so the set of possible characters that we can include. And now we have this special S that is the set of all possible strings given the alphabet. Okay. And now we define a function f that is taking two strings, okay, here for example S and T, and it outputs 0 or 1. 1 if S is a subset of T and 0 otherwise. In order to check if S is a substring of T, we need to compare S and T. A variant of this function that we want to implement is that f can take two strings and will output, for example, a natural number that will be the location of S in T. Let's imagine the first location, for example. Or it can even be another variant of this function that could output the locations, so all the possible locations of S in T. Now let's write a little example. Let's imagine that S is equal to A T, and let's imagine that T is a sequence with G, C, A, T, C, A, T. Now, if we apply F on S and T, if we consider the first case, in the first case, the output is going to be 1 because we have AT and AT here. In the second case, the output is going to be 1, 2, 3, 4 because AT, so S, is at position 4, the first one at least. And the third possibility here, the locations of S, it will be, the output would be 4, 5, 6, and 7. Okay, 4 and 7. Because S is here, S is here, it starts at position 4 and starts at position 7. Okay, so basically this is the idea of a string search algorithm. We are going to give to the algorithm 2 strings, and the idea will be to search one of the strings in the other string. And to do this, we need to, as I told you, compare S and T. Now, what does it mean to compare 2 strings? Let's first define this concept, comparing 2 strings. So if we want to compare 2 strings, it means that we want to quantify the similarity or difference, if you want, between both strings. And by similarity, we mean that we will need to define some kind of string metric or some kind of distance function. And in fact, there are different ways to measure the distance between 2 strings. For example, we have the Hamming distance. Very simple, very well known. We have other distances like the Levenshtein distance. And we can define other kinds of distances like the TF-IDF distance. Okay, so in this lecture we're mainly going to focus on the Hamming distance and the Levenshtein distance. In order to define the Hamming distance, we need to consider 2 strings, so S and T, for example, and we're going to compare the strings character by character. So let's imagine that S and T are the following strings. So this is for S, and for T we have this other string. Okay. What you can see here is that the strings should have the same size. So S is a string of size 4 with characters A, G, C, and T is a string with 4 characters as well, with characters A, G, C, and C. Now, the Hamming distance is going to compare the characters in a pairwise manner. So is A equal to A? Yes. Is G equal to G? Yes, it is. Is C equal to C? Yes, it is. Is T equal to C? No, it isn't. And we are simply going to sum the number of differences that we observe. So here, the only character that differs is T from C. So the distance is 1. If we want to define this distance in a more mathematical way, we need to define a function D that is going from our set of all strings to the set of natural numbers, for example. And in practice, it will be something like this. So we give the two sequences, and we are going to create a set with the positions I, such that for all I belonging to the set of natural numbers between 1 until the size of S, such that Ti is different from Si. So in this set we are simply keeping track of the positions where the two strings differ, and then we are simply computing the cardinality of this set, which is the number of difference that we have. So now let's study the Hamming distance in an example. So let's take S be A, T, T, G, C, for example, and we need to choose another sequence T that should have the same size. So for example, A, G, C, G, and T, for example. Okay, now we are going to apply this function here in order to compute the distance between T and S. So first, let's make this set, the inner set that is here, that simply contains the locations where T and S differ in a pairwise manner. So let's compare A and A. A is equal to A, so there's no difference here, so we are not going to keep track of position 1. T is different from G, so we are going to keep track of the difference. So this is position 1, 2, 3, 4, and 5. Position 2, we have a difference. Position 3, T is different from C, we have a difference. Position 4, we don't have a difference. And position 5, we do have a difference. So these are the locations where T and S differ from each other. Now we need to compute the cardinality of this, which is simply 3. So the Hamming distance between S and T is 3. Now we are going to also study during this chapter the Levenshtein distance, but this distance is a bit more complex, so we are going to do it in a different video. Now that we have studied this Hamming distance as an example of string metric, we can ask ourselves, why do we need to compare strings in bioinformatics? This is a very important question, and let's try to answer giving some possible applications of string search algorithms. So the first application could be, for example, genome assembly. Another possible application could be to identify mutations in genes or in whole genomes. We can also try to determine DNA regions correlated with a phenotypic trait. That is called, for example, QTL. This is an example for quantitative trait locus. We can also use it to study evolution. For example, the divergence of sequences or the speciation. And this is linked to the study of phylogeny. Which means to find the common ancestors between genes or between two species, for example. It can also be useful to identify mRNA alternative splicing. Could also be useful to quantify gene expression. And it could also be useful to identify patterns in proteins that will be responsible for some kind of function. So these are some of the examples. I will make small videos on these different examples that you can also check to give an idea how this comparison of strings will play an important role on these different applications. So first of all, let's try to, in a nutshell, explain what is genome assembly. So when we are going to, when we want to obtain the genome of an entire organism, for example, in different chromosomes, let's say, that's it. Each chromosome will be made of a sequence of A, T, G, C, for example, and so on. But we never have an entire lecture of an entire chromosome or all the chromosomes at once. When we try to obtain this genome of an organism, we usually start from reads, small reads. Let's imagine that our genome is this string. So this is my genome. Let's imagine that the alphabet contains all the possible letters, so this is not a real genome that we may observe, but this is just for the example. And let's imagine that we have a bunch of reads. For example, we have a first read, then we have another read, then another one, then another one, then another one. That's it. Let's imagine that we only have these reads. What we need to do with the reads is that we need to align the different parts of the reads. For example, we will align these letters together, and we will see that these two parts are connected. We're going to do the same with these other parts, and so on. And little by little, we are going to make the entire connections of the genome until obtaining each one of the letters of the genome, and so on. And to do this, we need to compare these different reads in order to see the portions that are in common in order to align them. That's why string search is useful for genome assembly. We also mentioned the application of determining mutations. In this case, let's imagine that we have genome 1 and genome 2. Let's imagine that genome 1 is the genome of reference, and genome 2 is some kind of variant, for example. Let's imagine that genome 2 is a variant that is responsible for some kind of disease, and we have the corresponding sequence. For example, this is my genome, and the variant is saying this as my genome. Now, if we want to determine mutations, we need to compare the different characters that are forming our sequences. Let's imagine that here, for this little example, we are using some kind of variant of the Manhattan let's imagine that for this example we are using the Hamming distance. So we are comparing character by character, and we see that here we have a difference, and otherwise we don't have any difference. So our mutation would be here. Of course, we are not going to use such a simple approach to determine mutations, but here is just an example to explain why string search will help in the determination of mutations in the analysis of genomes or genes. We also mentioned another possible application that could be on phylogeny. So in phylogeny, we will see that the link is quite straightforward. What we want to do in phylogeny is that, given the distance v between two genes or two genomes, for example S and T, in this case the genomes or the genes S and T will be two strings simply. Strings. When can we say that T and S are similar? So when can we say that between S and T there is some kind of relationship? And it's not any kind of relationship, but evolutionary relationship. Okay, is there a link between the two sequences? You can think regarding this example of the ship of Theseus, when you have the ship of Theseus, and you're replacing one part after the other. So you replace this little piece, for example, and then this other little piece breaks, so we need to replace it, and so on until we replace the entire ship. Can we say that the ship is the same or not? And the idea is similar in the case of two sequences. So we start from some kind of ancestral sequence, and we start like having mutations that accumulate differently in two other populations, two variants of the sequence, for example, and we may have some kind of speciation, or it could be the same sequence that is duplicated. So here we have some kind of duplication, and then we have accumulation of sequences, and the idea will be that after some time here we have SA and here we have SB or SA prime SB, and the idea is to see if we have any kind of relationship between the two, if at some point we have some kind of common ancestor. Okay, so shared ancestor or common ancestor. Okay, this is the main idea. Now before explaining this, let's give some definitions. So we will say that S and T are analogs if they are similar but they have separate evolutionary origins. So let's imagine that we have S, we have T here. They are similar, they look similar, but they don't have a common ancestor. It means that the similarity is not due to an evolutionary relationship, but it's just due to randomness. Okay. Then another definition is that S and T are homologous if their similarity is due to shared ancestry. Okay, so in this case it means that at some point S and T have had some kind of common ancestor. Let's call it R. Okay, so we had one common sequence R, for example. Then we had this duplication event and mutations, accumulations of mutations that lead to S and T. And this homology can be split in two variants. The first one will be orthology, when homology is due to speciation events. Okay, so in this part the important term here is speciation. And the other variant is paralogy, and paralogy is homology due to a duplication event. So the difference between paralogy and orthology is that orthology is due to speciation and paralogy is due to duplication. What does it mean? It means that, for example, for orthology, we have one species, for example S. So this is the gene of one species, of the common ancestor, and at some point some individuals, for example, were stuck in an island and start evolving separately from another set of individuals that were in the mainland. Okay, so let's imagine this scenario. We have the mainland and we have an island here. We have some of the individuals that are here, most of the other individuals are here, and we wait many generations. Mutations will accumulate and we have another species that appear, that is the green dots. Okay, and the other one has also evolved its way and now it's different from the ancestor one. So the ancestor is this one, and we have now two populations, 2 species. One is this one, and the other one is this one. So here we have a speciation. It means that if you take one individual from this one, this population, and another one from this population, they won't be able to give birth to an organism that can reproduce itself. Okay. While the duplication event is just, let's imagine you have the gene S. So we are dealing with one species, within the same species. And there is an event in the genome that is going to take all these parts and duplicate it and paste it here. So it's like a copy-paste. So after the copy-paste, we have just S. And let's imagine that the insertion is made here. So now we have a copy of S here. And after some generations, after some generations, some period of time, the 2 copies of S will accumulate mutations, and we will end up with S prime and S second. Okay. So this will be the duplication event. So S prime and S second are similar to each other because they share this common ancestor, but they have evolved differently after the duplication event. While here, for example, let's call this S O and S star. They evolve differently, not because we have 2 copies of S in the same genome, but because one population evolved separate from the other. But the result will be similar in any case. And you can have even phenomena where you have orthology and paralogy that start being intertwined. For example, you start with one sequence, S, for example, then you get a duplication, S and S prime, or S second and S prime, and then you have the speciation. So now we will have 2 variants, so we'll have S second 1, let's say, for the first species, S prime 1, S second 2, and S 2 prime. And you can have another event of duplication and so on, just to give an example. In this lecture, we have been studying string search algorithms, introducing the terms. We have defined what is a string, we have defined it as a sequence of characters, we defined what a character is, mainly through the alphabet, the set of all possible characters that may exist. We have also defined what is a sequence, what is a list, and what is a string finally. We have also provided these formal notations and an example for the DNA string. We have also defined what is an algorithm. We define it as a finite, unambiguous sequence of instructions. We have also defined 3 ways to characterize an algorithm, how well the algorithm is solving the task from the input to the desired output. And we have also characterized it in terms of time complexity. So how much time will the algorithm take with respect to some important variables? Here, for example, the length of the string. And the same thing for the space complexity, which means the amount of memory that the algorithm will require. We have also defined what is a string search algorithm. So we have defined it first through the definition of the alphabet, and also the set of all possible strings defined in the given alphabet. And then we have said that a string search algorithm is a function f that will take two strings and will output 0 or 1. So if we send two strings s and t to the function, it will output 1 if s is a substring of t and 0 otherwise. There are also some variants of this function. One of the variants could be, for example, to give the first location of s in t, or the other variant could be to give all the possible locations of s in t, as we have seen here. In order to search one string inside another string, we have seen that we need to compare the two strings. And to do this, we need to quantify the similarity between both strings. To do this, we need to define some kind of string metric or distance function. We have seen that there are many distance functions. We have mentioned 3 of them, and we have studied a little bit the Hamming distance, which is very easy to define. It only works for strings that have the same size. Here, for example, s and t both have a size of 4 characters. And the idea of the Hamming distance is simply to create the set of positions where the two strings differ. And then the distance is simply the cardinality of this set. Then we have studied different kinds of applications that may require to compare strings in bioinformatics. We have mentioned, for example, genome assembly, identification of mutations, the study of evolution, for example, it means phylogeny, and so on. Regarding these applications, we have focused on 3 applications. The first one was on genome assembly. We have seen how we can go from little reads to form the entire genome little by comparing the strings. We have also seen how string search can be used to determine mutations. And we have finally studied a little bit of phylogeny. We have defined some important terms, such as what are 2 sequences that are analogs, homologs, and within the homolog we have also seen the concept of orthology and paralogy, and how string search is important in order to find the relationship, the evolutionary relationship between sequences. And next time we are going to study other concepts, such as the Levenshtein distance that we have also mentioned today, and other kinds of ways to compare two strings.